One of the most eye-catching late summer and autumn mushrooms of northern forests coats itself in a golden, sticky glaze and dark scales, grows in dense clusters on living and deadwood, and recently revealed an extract dominated by linoleic acid that shows measurable antimicrobial and antibiofilm activity.
That is quite the introduction.
Right. I mean, it is a bold claim for a mushroom you might just walk right past on a hike. But welcome to the deep dive, everyone. Our mission today for you listening is to completely unpack the fascinating, totally hype-free reality of Pholiota aurivella.
Yeah, commonly known as the Golden Scalycap.
Exactly. And we are bypassing all those oversimplified, sometimes dangerously misleading popular field guides today. We're diving straight into the peer-reviewed morphological, chemical, and taxonomic studies, because we need to understand what this highly specialized ecological agent is really doing out there in the forest.
It's vital that we frame it that way, honestly. When people see this mushroom, they just see this photogenic pop of color on a log.
It is very pretty, yeah.
Right. But this organism completely challenges traditional taxonomy. I mean, if you look at its internal chemistry, it uses an array of secondary metabolites for survival that's just highly unusual for the fungal kingdom.
It's waging invisible wars out there.
Literally. It operates in a way that forces mycologists to question, long-held assumptions about how fungi defend their fleshy bodies in a really harsh, competitive, microscopic environment.
So before we can even get into that internal chemical warfare, we really have to understand the physical structure it presents to the world.
Yeah, because its appearance is notoriously tricky.
So let's paint a picture for the listener. What does Pholiota aurivella actually look like when you stumble on a fresh cluster in the woods?
Visually, a fresh specimen is striking. Macroscopically, you're looking at a cap that ranges from, about 5 to 15 centimeters in diameter.
Pretty decent size then.
Yeah, it's not tiny. When it first pushes out of the wood, it starts off broadly bell-shaped, like a tight little dome protecting the underside.
And then it opens up.
Right. As it matures over a few days, it takes on a plano-convex shape.
Meaning it flattens out but still keeps a curve.
Exactly. It flattens, but it usually keeps what we call a broad central umbo. It's a distinct raised bump right in the dead center of the cap.
Okay, got it. And the color is where it gets that Golden Scalycap name, right? It's not a subtle yellow.
Not at all. The ground color of the cap is this incredibly bright, saturated golden yellow.
Almost artificial looking.
Yeah, and it can deepen into this rich, tawny orange, especially right on that central umbo we talked about. It looks almost like a dollop of bright orange marmalade sitting on a log.
I love that visual.
But the cap is only half the story.
Yeah.
Underneath that cap is the hymenophore, the spore-bearing surface. And it features crowded, adnate gills.
Adnate meaning how they attach to the stem.
You got it. Adnate gills are attached broadly and directly to the stem, without dipping down or pulling away.
What color are they?
When it's young, they start off pale cream, almost white. But as it matures, and the millions of microscopic spores develop, they darken significantly.
To what?
They shift into a deep, rusty cinnamon brown.
Oh, wow. That's a big shift from white.
It is, and that color change is entirely driven by the spores themselves. If you took the cap, cut off the stem, and laid it on white paper overnight, it would drop a massive spore print that is a distinct cinnamon brown color.
Okay, so we have the golden cap, the rusty gills, and holding this all up is a pretty substantial stem, right?
The stem, or the stipe, can be anywhere from 3 to 15 centimeters long. It really depends on how far it had to stretch to reach the open air from inside the wood.
Right, because it starts deep inside the log.
Exactly. And the texture of the stem changes dramatically. Near the top, just under the gills, it's pale yellow and relatively dry. But as you move down toward the base, it's covered in these recurved, shaggy brown scales.
They almost look like little peeling shingles on a roof.
That's a perfect way to describe it. And dividing that smooth top from the scaly bottom is an ephemeral, flimsy ring zone.
Left by the partial veil.
Right, a yellow fibrillose partial veil.
Let's break down what a partial veil actually is for the listener, because it's not just a decorative skirt on the mushroom, it's serious fungal engineering.
It really is. A partial veil is a delicate web of tissue that stretches from the edge of the cap to the stem when it's very young.
To protect the gills.
Exactly. Its evolutionary purpose is to create a sealed, humid microchamber. It keeps the developing gills from drying out and shields them from hungry insects.
But then the cap expands.
Right, as the cap flattens into that plano-convex shape, the veil stretches until it finally tears. Once it breaks, the remnants hang onto the upper stem, leaving that faint ring. but it's so flimsy it usually weathers away in a few days.
Okay, so we've got the golden cap, the dark gills, the shaggy stem. But here is where the shape-shifting deception comes in.
Ah, yes, the slime.
Right. We have to talk about the slime and the scales, because this is where those glossy field guides completely fail casual observers.
They really do. So that golden ground color is covered by a highly viscid, gelatinous pellicle.
A pellicle is just a scientific word for a slimy skin, basically.
Essentially, yes. It's a thick, removable, slimy outer skin. And sitting suspended inside that slimy matrix are flat, sometimes slightly upturned, dark, reddish-brown scales.
So the textbook image is this bright gold mushroom covered in sticky slime, speckled with contrasting dark brown scales.
Exactly.
But those scales are basically a magic trick. If you're a casual forager, you go out with your guidebook, you see a pristine photo of a golden mushroom with dark scales. But if it rained heavily the night before...
Which is super common in autumn.
Right. You might walk right past Pholiota aurivella because the heavy rain interacts with that slime.
It dissolves it entirely. The rain washes away the gelatinous matrix. And because the scales are just suspended in that slime, they wash right off with it.
Just completely gone.
Yep. Sometimes they melt down into these indistinct smudges you barely notice. What you're left with after heavy rain is a completely smooth, glossy, varnished-looking yellow cap.
No scales at all. That is incredibly frustrating.
It's a taxonomist's nightmare in the field.
It's like trying to identify a leopard that sheds its spots when it gets wet. If the defining physical features just melt in the rain, how do scientists actually identify it? If I send you a photo of a smooth yellow mushroom on a log, how do you know?
The harsh reality is, in the field, with just the naked eye, you often don't know. You can't be scientifically certain.
Which is the fatal flaw of static field guides.
Exactly. They capture one idealized moment. True scientific identification requires bringing it back to the lab and putting it under a microscope. You have to look at the structural biology that rain cannot wash away.
We have to zoom in. So let's talk about the microscopic truth. What are we looking for under the lens?
We always start by measuring the spores. Pholiota aurivella produces smooth ellipsoidal spores.
Ellipsoidal meaning shaped like tiny footballs.
Right. But crucially, they are thick-walled, built like little armored capsules. And they measure consistently between 7.5 and 11 micrometers long and 4 to 8 micrometers wide.
And they have a very specific escape hatch built in, don't they?
They do. They feature a distinct apical germ pore. If you look at the tip of the spore under high magnification, you'll see a small, specialized thinning of the cell wall.
Like a little trap door.
Exactly. When the spore lands in a suitable environment, like a damp crack in wood, the new fungal hyphae pushes its way out through that specific germ pore. It's a localized weak point designed for emergence.
What about the tissues of the mushroom itself, the actual flesh?
When you slice the tissue extremely thin and stain it, you look for two major things. First are clamp connections.
Oh, I love clamp connections.
Yeah.
They look like a microscopic aqueduct system. Let's explain them to the listener because they're wild.
They really are. A clamp connection is a small, semicircular, bridge-like structure that forms between two adjacent cells in a fungal thread.
Why do they need a bridge?
Because when a fungal cell divides, it needs to make sure a copy of each of its two different nuclei ends up in the new cell. The clamp connection acts as a literal bypass tube.
To get around the wall.
Right. It allows one of the dividing nuclei to detour around the cell wall barrier and drop safely into the new compartment, ensuring genetic stability.
It's an elegant plumbing system.
And its presence is a massive clue for taxonomists. The second thing you look for are specialized cells on the gills called cystidia.
Sterile cells, right. They don't make spores.
Correct. Scattered among the spore-producing basidia are these sterile cells. In Pholiota aurivella, we look for chrysocystidia.
Chrysocystidia, what makes them special?
When stained with alkaline solutions, they contain a yellowish, light-refracting body inside them. We also look for chylous which are clustered on the very edges of the gills. The exact shape and chemical reactivity of these cells are the definitive fingerprints of the species.
It's just amazing that we go from looking at a bright yellow slimy cap in the woods to meticulously measuring tiny bypass tubes and light refracting cells in a lab.
It's the only way to be sure.
But all of this, the thick-walled armored spores, the slimy cap, the robust stem, none of this is just for show. These are highly specific adaptations for surviving in a brutal ecological niche.
Absolutely brutal.
Let's move into the forest pathobiome because this organism's entire existence revolves around one monumentally difficult task: eating solid wood.
Which is a formidable biochemical challenge. We just look at a tree trunk and see wood. But on a molecular level, wood is a fortress.
Right, it's mostly cellulose and lignin.
Exactly. Cellulose provides the tensile strength, and lignin is this highly complex, chaotic, three-dimensional polymer that acts like biological concrete holding everything together.
And lignin is tough. Most things on Earth can't digest it.
It is incredibly degradation resistant, but Pholiota aurivella can eat it. And to do that, it plays a very strategic long game. It operates initially as a weak, wound-associated parasite.
When you say weak, you mean it's not aggressively taking down a healthy tree?
Right. It doesn't attack an intact tree with thick bark. It waits for an opening. A branch breaks in a storm, a frost cracks the trunk, an animal gouges the bark.
And a spore just happens to land there.
A wind-blown spore lands in that damp wound, germinates, and the fungal mycelium quietly infiltrates the living tree.
Sneaking in through the back door?
Precisely. Once inside, it slowly colonizes the heartwood, the dense inner core. The tree is still alive, still making leaves, but the fungus is inside, slowly compromising the structural integrity over years or decades.
Just biding its time.
Until the inevitable happens, the tree dies, or maybe that structural weakness causes it to snap in high winds. And the moment that host tree dies, the fungus shifts gears completely.
It goes from parasite to saprotroph.
Yes, a saprotroph feeds on dead organic matter. Without the tree's active immune responses fighting back, the fungus goes into overdrive, rapidly recycling that massive accumulation of dead wood back into the soil.
Does it care what kind of tree it's eating? Because some fungi are so picky, like they'll only eat one specific type of oak.
It has preferences, but it's remarkably adaptable. Across the northern hemisphere, its primary hosts are deciduous hardwoods. It shows a really strong affinity for Fagus beech trees.
Okay, beech.
But you'll also find it extensively on maple, birch, elm, ash, poplar, and willow.
That's a huge menu of hardwoods. But hardwoods have a specific lignin structure.
They do, and you'd think its tools would be locked into that structure.
Yeah.
But here's the fascinating part. In montane regions, higher mountain elevations, and in western North American forests, it adapts perfectly well to coniferous hosts.
Softwoods, like pine.
Yep, pine and fir, which have fundamentally different lignin chemistry. It's just a highly versatile ecological recycling engine.
So it's inside the log, whether it's a beech in New York or a fir in Oregon. How does it physically eat the tree? Because it's categorized as an active agent of white rot.
Right. To understand white rot, you have to look at the two broad categories of wood decay fungi,
Yeah.
brown rot and white rot.
Let's start with brown rot.
Brown rot fungi only have the enzymes to break down cellulose. They extract the carbohydrates, but they can't break down the tough lignin, so they leave it behind. If you've ever seen a log that breaks apart into dry, brown, crumbly cubes, that's brown rot.
Looks like crumbling brown bricks.
Exactly. But Pholiota aurivella is a white rot fungus. It has the advanced biochemical machinery to completely degrade that highly resistant lignin right alongside the cellulose.
It strips everything down at the same time.
Yes. The result is it turns the solid wood into this soft, spongy, fibrous white mass. The white color is actually the bleached cellulose fibers remaining as the dark lignin is actively stripped away.
I want to focus on that biochemical machinery because how it does this is staggering. It's not chewing the wood. It's basically turning the inside of the tree into a highly localized acidic chemical refinery.
It's an extreme environment. The fungus secretes a barrage of primary extracellular enzymes directly into the wood.
Extracellular meaning it pumps them outside its body to digest food before absorbing it.
Right. Let's break down the three heavy hitters in this enzymatic arsenal, starting with lacase.
Okay, lacase.
Lacase is a copper-containing enzyme. Its main job is to oxidize phenolic compounds in the wood. What's incredible about lacase in this specific mushroom is its optimal operating environment. Its activity peaks at a highly acidic pH of 3.0.
Wait, a pH of 3.0. For you listening, neutral water is 7.0. Black coffee is around 5.0. A pH of 3.0 is basically the acidity of concentrated vinegar or grapefruit juice. The fungus is acidifying the wood just to make the enzyme work better.
It relies on that acidity, and it gets more intense with the second enzyme, manganese-dependent peroxidase, or MNP.
MNP, what does that do?
It's a heme-containing enzyme, meaning it uses an iron core, like our hemoglobin. But MNP takes naturally occurring divalent manganese ions in the wood manganese with a +2 charge and oxidizes them into trivalent manganese, which has a +3 charge.
Whoa, hold on. We're throwing some heavy chemistry around. Divalent to trivalent. Why does the fungus care about the electrical charge of a manganese atom?
Because trivalent manganese is a highly reactive, unstable free radical. It desperately wants to steal an electron to stabilize itself.
So it becomes destructive.
Exactly. And because it's so small, this trivalent manganese ion can diffuse deep into the microscopic pores of the dense lignin polymer, places the larger enzymes literally can't fit.
Like a tiny saboteur?
Once inside, it acts like a tiny chemical buzzsaw. It violently tears electrons away from the structural bonds of the lignin, causing the whole molecular structure to crack and fall apart.
And what's the pH for this buzzsaw?
MMP hits its absolute peak efficiency at an acidic pH range of 3.0 to 4.5.
So it's sending free radicals into the crevices. What's the third enzyme?
Lignin peroxidase.
Yeah.
While MNP uses that manganese mediator, lignin peroxidase directly attacks the non-phenolic parts of the lignin structure. And this one demands an even more extreme environment. Its activity peaks at a pH of 2.0 to 4.0.
A pH of 2.0, that is equivalent to human stomach acid.
It is intensely corrosive.
The inside of that rotting log is basically a vat of highly corrosive acid, swarming with free radicals, tearing apart the toughest plant matter on Earth. It's a miracle the fungus doesn't dissolve its own mycelium.
It must have incredible proton pumps in its cell walls just to survive the environment it creates. It's a masterclass in extreme biological adaptation.
But here's the paradox. Despite this incredibly harsh, acidic, hostile environment inside the wood, the mushrooms popping out into the air serve as a crucial habitat for other organisms.
Right, it's an ecosystem within an ecosystem. We have to talk about the pleasing fungus beetle.
Triplax californica. I genuinely love that common name, the pleasing fungus beetle. It sounds so polite.
Well, it's descriptive of the entire family of Arotilidae beetles. These beetles have evolved a life cycle tied intimately to fungi, like Pholiota aurivella. They aren't deterred by the slime or the chemical defenses.
Yeah, thrive on it.
They do. The adult beetles and their larvae live in and feed directly upon the fleshy fruiting bodies and the gills of the mushroom.
So they're eating the reproductive organs of the fungus. That sounds entirely parasitic. How does the mushroom benefit from this at all?
It's a trade-off that borders on mutualism, actually. The beetles get a reliable food source and a sheltered place to lay eggs. In return, as they crawl through the sticky gills, they get covered in thousands of those thick-walled spores.
So they become delivery drivers.
Exactly. When they fly off to find another cluster or a newly fallen tree, they carry the spores. They transport them to new habitats much more precisely than the wind ever could.
That's incredible. So we've established a lot here. We know it's widely distributed. It eats everything from beech to fur. It drops its scales in the rain. It builds acid refineries.
So the busy organism.
But because of this wide distribution and shape-shifting nature, historical mycologists really struggled to categorize it. You can imagine a naturalist in the 1800s seeing a slimy, yellow, scaly mushroom eating a dead tree and just assuming any mushroom doing that job was the exact same species.
And they did assume that.
But modern DNA and mating studies tell a radically different story. Let's get into the taxonomic tangles because things get incredibly messy here.
The taxonomy is a perfect, almost frustrating example of how mycology has evolved. For a very long time, Pholiota aurivella was lumped into a massive bucket called the section adiposi complex, or the Pholiota adiposa group.
And a lot of popular field guides still perpetuate this confusion today, don't they?
Oh, constantly. They frequently merge three entirely distinct biological species, Pholiota aurivella, Pholiota laminella, and Pholiota adiposa under one common name. Or they just treat them as interchangeable.
Why do they do that? Just for simplicity?
Primarily because, to the naked eye in the woods, their morphological overlap is massive. They are all essentially golden yellow, slimy mushrooms with dark scales growing in tufted clusters on dead wood.
Right, without a microscope, separating them is just guesswork based on slight variations in color or slime, which we know change with the rain anytime.
Exactly, but under the microscope, the truth comes out.
Back to the spores.
Yes, the microscopic separation is clear if you meticulously measure them. Pholiota aurivella is the heavyweight. It has the largest spores, 7.5 to 11 micrometers long. Pholiota laminella has significantly smaller spores, maxing out around 6.0 to 7.5 micrometers, and Pholiota adiposa sits somewhere in the middle.
But for decades, scientists must have argued about this. Spore size seems like a small detail to split a species over. You could argue maybe it's the same mushroom, but the ones on better wood just make slightly bigger spores.
That was exactly the debate. And it brings us to the gold standard of biological species definition, the mating compatibility studies.
This is how they finally proved they were looking at different biological entities. Let's explain how you mate fungi in a lab, because it's not like putting two mice in a cage. We're talking about the historical mating trials by Farr and colleagues.
Right. To do a mating study on fungi, you first have to grow what's called monokaryotic mycelium. You take a single microscopic spore from a mushroom and let it germinate in a petri dish full of agar gel.
Okay, one spore.
Because it came from one spore, the resulting fungal network only has one set of genetic information. It's essentially a half-organism waiting for a partner.
It's swiping right on Tinder, waiting for a match.
In a highly sterile biochemical way, yes.
Yeah.
So you take the monokaryotic mycelium from mushroom A and put it in the same dish as the monokaryotic mycelium from mushroom B. The two networks grow toward each other.
And when they meet,
If they recognize each other as the same biological species, the cells will physically fuse together. They exchange genetic material, create those clamp connections we talked about, and form a dikaryotic mycelium, a fully complete organism capable of producing a new mushroom.
and if they aren't the same species,
They absolutely refuse to fuse. They might grow right past each other or form a barrier, but they will not exchange genetic material. They are genetically isolated.
Wait, how does a fungus even know it's incompatible? It doesn't have a brain. How does a string of cells in a dish know the cells next to it are a limonella and not an aurivella?
It all comes down to incredibly complex chemical signaling. They excrete pheromones and possess highly specific receptor proteins on their cell walls.
Like a lock and key system.
My microscopic lock and key. If the pheromone from one doesn't perfectly fit the receptor of the other, the biochemical cascade required to merge the nuclei simply never triggers.
That is mind-blowing. So what did Farr actually find when he put all these look-alike mushrooms into the petri dishes?
Farr's work was foundational. He gathered samples of various visually distinct North American species that older mycologists had given a whole host of confusing names. Names like Pholiota aviatus and Pholiota cannata.
Okay, so a bunch of different names.
Right. He put them in the dishes and proved they were completely interfertile. They readily mated with each other. Because of that, he determined all those different names were just visual variations of the same organism. He rolled them all up into one valid species, Pholiota limonella.
He cleaned up the messy naming system.
He did. But the crucial paradigm-shifting discovery happened when he tried to cross that newly defined limonella group with samples of true Pholiota aurivella.
Yeah.
They were entirely 100% incompatible. They flat out rejected each other. They cannot mate under any circumstances.
Proving definitively that despite looking like identical twins, they are distinct biological species.
With strict reproductive barriers, it completely validated the microscopic spore measurements.
So if they can't mate, they're different species. Case closed. But science rarely stops at case closed. How does modern genetics complicate this even further? Because we aren't just relying on microscopes and petri dishes in the 21st century.
Modern genetics always adds a layer of beautiful, frustrating complexity. Today, mycologists utilize ITSS ribosomal RNA sequencing.
Let's ground the listener here. What is ITSS and why do we sequence ribosomes?
IT stands for internal transcribed spacer. Think of it as the universal barcode of life for fungi.
Okay, a barcode.
It's a specific, highly variable region of DNA situated right between the genes that code for ribosomes. Now, ribosomes are the cellular factories that make proteins, so the genes that build them are highly conserved. They don't change much over millions of years because if you break the factory, the cell dies.
Makes sense.
But the ITS region, the spacer DNA between those important genes, mutates rapidly and freely because it doesn't code for critical structures. By reading the exact sequence of letters in that spacer region, scientists can tell exactly how closely related two fungi are.
It's like looking at the typos in a manuscript to see if two copies came from the same author. So what did the DNA barcodes reveal about our golden mushroom?
The DNA showed that this is a highly variable, globally scattered phylogenetic group. Sequencing revealed a massive genetic cluster historically called the Adipocella-Monella-Aurivella complex. But sitting right next to it on the evolutionary tree, genetically distinct, is a sister clade of true Pholiota aurivella.
And geography plays a massive role in this genetic mapping, doesn't it? An ocean is a pretty effective barrier against spore dispersal.
Absolutely. Geography and deep time matter immensely. For instance, there are specimens collected in New Zealand that for decades were assumed to be Pholiota aurivella.
They looked identical.
Macroscopically, they looked identical to the ones eating beech trees in Europe and North America. Under a microscope, their spores looked right. But when their ITS barcodes were finally sequenced, they were found to be genetically distinct.
Wow.
They formed their own separate isolated group. Millions of years of geographical isolation allowed their DNA to drift apart, even while maintaining the exact same physical appearance.
So what looks like one species to a forager's naked eye is actually a hidden web of genetically isolated, globally distributed cousins.
Exactly.
Okay, so we've established how it breaks down wood with a localized pH 2.0 acid refinery, and we know how strict its genetic boundaries are. But this brings us to a burning question, maybe the most important question of all.
How does it defend itself?
Yes. Think about it. This fleshy, highly nutritious fruiting body is pushing its way out of a damp, rotting log. It is bathed in moisture, surrounded by an absolute swarm of bacteria and microbes that thrive and decay. By all logic, this mushroom should be instantly devoured by bacterial rot the moment it hits the air. How does it protect its fleshy tissues?
It's the perfect question. And the answer represents a completely divergent evolutionary path that stunned researchers. First, we have to look at how most fungi defend themselves.
What's the standard armor?
For the vast majority of macrofungi, chemical protection comes from polar phenolic compounds.
What are phenolic compounds?
They are complex, ring-shaped molecules that act as powerful antioxidants. In that chaotic oxidative environment of a rotting log, free radicals are constantly threatening to tear cellular tissues apart.
Like the trivalent manganese we talked about earlier.
Exactly. Phenolic compounds act like molecular sponges. They scavenge free radicals, neutralize them, and protect the tissues from oxidative stress and microbial attack. It's the standard issue armor of the fungal world.
But not Pholiota aurivella. It threw that armor away.
It did. This is what's so fascinating. We have a milestone 2025 study by Yaman and colleagues. They used a technique called GCMS gas chromatography mass spectrometry.
Let's explain GCMS. It sounds intimidating, but the concept is actually brilliant.
It really is. Essentially, you take an extract of the mushroom, heat it up until it vaporizes into a gas, and shoot that gas through a long, coiled microscopic tube.
And the molecules race through the tube.
Right. Different molecules travel through the tube at different speeds depending on their chemical properties. That's the chromatography part. As they exit, they get hit with an electron beam that shatters them into pieces, and a magnetic field measures the mass of those pieces. That's the mass spectrometry part.
Giving you a highly precise fingerprint of every single molecule inside the mushroom.
Precisely. And what that fingerprint revealed was shocking. Pholiota aurivella has entirely abandoned the standard phenolic defense strategy. The researchers found 0 phenolic compounds in its extract. Absolutely none. They tested its antioxidant capacity and found it to be exceptionally low. According to traditional fungal biochemistry, this mushroom should be utterly defenseless.
So if it has 0 phenolics, what did it replace the armor with? How is it surviving?
It replaced the armor with a biological weapon. It uses a lipid defense system.
Lipids, so fats.
Yes. The GCMS showed its ethanol extract is completely dominated by lipid-derived compounds. Specifically, nearly 60%, 59.2% to be exact, is polyunsaturated linoleic acid, alongside about 17% ethyl linoleate. It has evolved to weaponize fats.
Weaponized fat. Okay, I need to understand the mechanics of this because it sounds crazy. How does a simple fat like linoleic acid fight off a bacterial infection?
It comes down to the physical geometry and chemistry of the molecules. Free fatty acids like linoleic acid act as natural surfactants. Let's think about a bacterial cell membrane.
Okay, the bacterial force field.
Right. To survive, a bacterium has to keep its internal machinery separated from the outside world. It uses a delicate lipid bilayer, a microscopic double-layered sandwich of fats that creates a flexible skin.
Got it.
Now, linoleic acid has a very specific shape. It's a long hydrocarbon chain with a couple of kinks in it caused by double bonds. Because it's also a lipid, it has a chemical affinity for the bacterium's lipid bilayer.
So it's drawn to it.
Yes. The linoleic acid from the mushroom physically inserts itself, wedging its long, kinked structure right into the tight, orderly sandwich of the bacterial membrane.
It infiltrates the force field.
It infiltrates it, and because of its kinky shape, it completely disrupts the structural integrity of the membrane. It forces the bacterial lipids apart. It creates gaps.
And what happens to the bacteria?
This causes immediate intracellular leakage. Vital ions and cellular contents just spill out into the environment. It essentially bleeds the bacteria out, destroys the electrical gradients required for energy, and completely stops cellular respiration. The bacteria suffocates and dissolves.
That is brutal. It's basically like popping a microscopic water balloon with a pin. Or an even better analogy, it's like using dish soap to break apart thick grease in a frying pan.
That is a highly accurate analogy.
The grease is holding together, but you drop the soap in, the surfactant breaks the surface tension, and the grease just shatters. The mushroom is literally synthesizing a biological detergent to tear apart the cell walls of invading bacteria.
It is a biological detergent.
Yeah.
And the 2025 study didn't just identify this in a vacuum, they proved its real-world efficacy. They tested this lipid extract against 16 different microorganisms.
And what did they find?
They found measurable, dose-dependent antimicrobial activity.
Dose-dependent meaning the more extract you use, the more bacteria die.
Exactly. They measured it by creating zones of inhibition, placing a drop of extract on a petri dish covered in bacteria and measuring the clear, dead circle that forms around the drop.
And it wasn't just weak environmental bacteria.
What were they testing it on? It created expanding zones of inhibition against serious clinical, multi-drug resistant pathogens. Severe human pathogens like Klebsiella pneumonia and Enterobacter aerogenes. The mushroom's biological detergent killed them.
And the craziest part of that study is that it didn't stop at free-floating bacteria. It also attacks biofilms.
Yes, which from a medical and ecological standpoint is arguably more impressive. Biofilms are the ultimate bacterial defense.
When bacteria hunker down.
Right. When bacteria realize they're under attack, they stop floating freely. They anchor to a surface and secrete a thick, protective, slimy matrix over the entire colony. It's a microbial fortress. Biofilms are notoriously hard to break down. They repel antibiotics.
Plaque on your teeth is a biofilm, right? It's incredibly stubborn.
Exactly. But this lipid extract actively inhibits the formation of biofilms, showing its strongest disruptive effects against pathogens like Listeria inaqua and Bacillus subtilis.
Think about the profound implication of that. A simple wood-decaying mushroom has evolved a targeted, lipid-based countermeasure to defeat one of the most stubborn defensive structures in the bacterial world, completely abandoning traditional antioxidants to do it.
It's an evolutionary marvel.
Okay, so the lipids act as this blunt force biological detergent, popping bacterial membranes. It's a localized carpet bombing of fatty acids. But Pholiota aurivella isn't just relying on blunt force, it also produces highly specific, targeted proteins for complex interactions. The chemistry goes even deeper. We have to talk about the carbohydrate decoder, PAA.
PAA stands for Pholiota aurivella agglutinin. This takes us completely away from the broad-spectrum lipids and drops us into the highly precise world of lectins.
This discovery goes back a bit further than the 2025 lipid study, right? This was a major milestone back in the 90s.
A massive breakthrough, a landmark 1991 study by Kawagishi and colleagues. They were the first to isolate this novel lectin from the fruiting bodies, and they did it using a technique called preparative PAGE.
Polyacrylamide gel electrophoresis. For anyone who has never worked in a lab, how do you pull a single invisible protein out of blended-up mushroom soup?
It's essentially a microscopic obstacle course. You create a thick slab of polyacrylamide gel, which looks like a dense, tangled sponge or highly concentrated jello.
Okay, a slab of jello.
You take your mushroom extract, place it at the top of the gel, and apply a strong electrical current across the slab. Because proteins carry electrical charges, the current physically pulls them through the gel.
But the gel is an obstacle course.
Right. Smaller proteins navigate the holes quickly and race to the bottom, while massive, bulky proteins get tangled up and move very slowly, staying near the top. By cutting the gel into horizontal slices, you can cleanly separate proteins based entirely on size.
So Kogishi runs the jello obstacle course and manages to isolate this specific protein, PAA. Why was this such a big deal in 1991?
Because it was the very first lectin ever characterized in the entire Strophareaceae family of fungi. It opened up a whole new understanding of their biochemical complexity.
So what exactly is this lectin structurally?
He found that it is absolutely massive. PAA is a high molecular weight polymeric protein. It isn't just one simple string of amino acids, it's a huge complex structure made of repeating parts, totaling over several hundred kilodaltons.
That's huge for a protein.
But the elegant part is that this massive structure is built entirely out of identical, smaller 18 kilodalton subunits, snapping together like identical Lego bricks to form a massive tower.
Wow.
They even meticulously mapped the exact sequence of amino acids at the starting end of these subunits, a highly specific chain of 16 amino acids starting with tyrosine and serine.
Okay, so we have this massive identical subunit protein Lego tower. But structure only matters if it does something. What is its function? What does a lectin actually do?
A lectin is a specialized protein that binds to specific carbohydrates, meaning complex sugars. PAA functions by agglutinating, which is the scientific term for aggressively clumping together human red blood cells.
It clumps human blood.
Yes. And what's fascinating is it does this uniformly across all human blood types. Type A, type B, AB, type O, it doesn't matter.
Yeah.
It clumps them all with equal ferocity.
That's aggressive.
And they found that if they chemically treated the red blood cells beforehand with an enzyme called pronase, which strips away some of the outer proteins on the cell surface, the blood cells became even more wildly sensitive to this clumping.
But the specificity of what it binds to is the real key to understanding lectins, isn't it? It's not just a sticky ball of glue.
Right. Think of a lectin as a piece of molecular Velcro, but it's a very specific type of Velcro that only attaches to one exact shape of lint.
Okay, I like that.
When scientists test lectins, they try to inhibit the clumping action. They flood the environment with different sugars to see which one the lectin prefers. If the lectin binds to the free-floating sugar, it stops clumping the blood cells.
It's like throwing the perfect shape of lint onto the Velcro so it can't stick to anything else anymore.
Precisely. Now, if you flood PAA with simple sugars, common things like glucose, fructose, mannose, it does absolutely nothing. PAA completely ignores them. The Velcro remains sticky.
It wants something specific.
The researchers discovered that PAA is only inhibited by a very complex, specific glycoprotein called acylfatuin.
Acylfatuin. That is a mouthful. What does that actually mean in biochemical terms? What is PAA looking for?
It means PAA has a highly specific, intense binding affinity for complex branched glycans. Deserlatrated just means a specific terminal sugar called sialic acid has been removed, exposing the complex branching structure underneath.
It's not looking for simple fuel.
No. It's looking for a very specific, highly complicated molecular keyhole to fit into on the surface of foreign cells.
Which raises an incredibly important question about identity. We spent time earlier talking about how hard it is to tell Pholiota aurivella apart from its close cousin, Pholiota adiposa. Do they have the same proteins inside? Does adiposa have this massive PAA Velcro too?
Not even close. And this is further proof of how fundamentally distinct these organisms are. Pholiota adiposa produces its own lectin, appropriately named PAL.
And how does PAL compare to PAA?
The biochemical differences are stark. First, PAL is tiny. It's only 32 kilodaltons, a fraction of the size of PAA.
Does it bind to the same complex sugars?
No. PAL is easily inhibited by inulin, which is a relatively simple plant carbohydrate. It doesn't require that complex acylfatuin keyhole.
Okay.
And most critically, PAL shows potent anti-proliferative activity against tumor cells, and it actively inhibits the HIV-1 reverse transcriptase enzyme.
Wait, really?
Yes, but PAA exhibits absolutely none of those pharmaceutical activities.
That is staggering. Outwardly, these two mushrooms look like identical twins. Golden, slimy, scaly, growing on the exact same logs. But internally, on a molecular level, they are operating with entirely different biochemical toolkits. One is wielding massive blood-clumping Velcro, while the other is wielding tiny proteins that inhibit viral enzymes.
It completely shatters the illusion that physical similarity implies biochemical similarity. The fungal kingdom is incredibly deceptive.
Speaking of deception, we really need to address the elephant in the room here. Knowing all of this, knowing that this mushroom is packed with membrane-bursting, cell-disrupting fatty acid detergents, powerful blood-clumping lectins, and lives bathed in a localized vat of pH 2.0 acid to digest solid wood, the inevitable question for any forager listening is, should you eat it?
The tox pharmacology of this species is highly contentious in historical foraging literature. You will find a lot of conflicting advice in old books.
Right, because there's this infamous culinary myth surrounding it that refuses to die. David Aurora, in his classic 1986 field guide Mushrooms Demystified, described the taste and texture of Pholiota aurivella as marshmallows without the sugar.
Which is a phrase that has caused a lot of stomach aches over the years.
Exactly. It sounds almost inviting. You read marshmallow and you think, I should toss that in a stir-fry. Although, to be fair, other foragers have reported a distinctly bitter or even a sharp metallic taste, which probably speaks to regional chemical variations. But still, the phrase sugarless marshmallow makes people want to try it.
It does, and that is a dangerous temptation. Modern scientific literature across the board strongly advises against human consumption of Pholiota aurivella.
Let's be specific. What actually happens to a person if they ignore the science, fry up a pan of these slimy mushrooms, and eat them?
Ingestion frequently leads to acute gastrointestinal distress, and I don't mean a mild upset stomach. We're talking about severe nausea, debilitating abdominal cramping, violent vomiting, and diarrhea.
How fast does it hit?
The onset is usually quite rapid, appearing anywhere from 30 minutes to 3 hours after consumption. It's a profoundly unpleasant, medically concerning experience.
Do we know exactly what is triggering the illness? Is it the massive dose of linoleic acid detergent tearing at our own cell linings, the lectins?
The precise molecule hasn't been definitively isolated as a single toxin, but the current scientific consensus strongly points toward irritants that are highly concentrated specifically in the gelatinous pellicle, that thick, sticky, slimy layer on the cap.
Which leads to another dangerous piece of historical foraging lore.
Yes. In older foraging circles, there was this persistent belief that if you simply sat down and painstakingly peeled that slime layer completely off the cap before cooking, the mushroom would be perfectly safe.
But that's not true.
I cannot stress this enough, that historical practice is scientifically unvalidated and unsafe.
Okay.
You cannot guarantee you've removed all the microscopic irritants embedded in the cellular tissue. Considering the severity of the gastric distress, it is simply not worth the risk. Why try to eat a biological detergent?
Exactly. But the stomachache isn't even the scariest part. Beyond its own internal toxicity, there is an exponentially bigger danger for foragers.
Misidentification.
Right. Pholiota aurivella shares its habitat-decaying wood and its general appearance with some truly nasty, life-threatening characters.
Relying on that golden color and scaly pattern can lead you to disaster. It has several lookalikes that you absolutely do not want to cross paths with. First, there's a sister species in the exact same genus, Pholiota squarosa.
If I'm looking at a log, how do I tell squarosa apart from aurivella?
Pholiota squarosa has a completely dry cap, not a slimy one, and its scales are not flat and suspended in gel. They are sharp, rigid, prominent, and recurved. They feel almost prickly.
And what happens if you eat it?
It causes severe gastrointestinal poisoning. But uniquely, that poisoning is exponentially magnified if you consume it with alcohol. It triggers a highly specific, severe physiological reaction.
That is a terrible Friday night, frying up mushrooms with a glass of wine and ending up in the emergency room. What else is out there pretending to be our golden mushroom?
Then you have Gymnopolis junoneus, widely known as the laughing gym. This is a very large, clustering, vibrant golden-orange mushroom that also fruits magnificently on deadwood. From a distance, a cluster looks remarkably like Pholiota.
But the internal chemistry is wildly different.
Completely different. The laughing gem is intensely, unpalatably bitter. But more importantly, it contains potent neurotoxic and hallucinogenic compounds, specifically psilocybin and gymnopolins. Mistaking aurivella for the laughing gem will result in profound, involuntary neurological and hallucinogenic effects.
That's bad, but it's not lethal. The most terrifying lookalike, the one that makes foraging for small brown or yellow mushrooms on wood so inherently dangerous, has to be the deadly Galerina.
Yes, Galerina marginata.
Even the Latin name sounds ominous. Marginata.
It is arguably one of the most dangerous mushrooms in the forest. This is a small, brownish-yellow mushroom that also clusters heavily on decaying wood in the exact same habitats. You might hear a small brown mushroom and think it doesn't look anything like a bright golden scaly mushroom, but we have to remember the shapeshifter nature of Pholiota.
The rain washing the spots away.
Exactly. If a young, tightly clustered group of Pholiota aurivella buttons gets heavily rainwashed over a few days, losing their distinctive dark scales and having some of their bright golden pigments bleached out, they look incredibly dangerously similar to Galerina marginata.
And Galerina contains lethal amatoxins. Let's explain what amatoxins actually do to the human body, because it is horrifying. It's not just a toxin that makes you throw up. It is an assassin at the cellular level.
It is a molecular assassin. Amatoxins specifically target and bind to an enzyme in your cells called RNA polymerase II. This is the enzyme responsible for transcribing DNA into messenger RNA. In simple terms, it's the enzyme that tells your cells how to build the proteins they need to survive.
So it stops that process.
Amatoxins permanently jam that enzyme. It halts all cellular protein synthesis.
So the cells simply stop functioning and die.
Yes, because your liver and kidneys are the primary organs trying to filter the blood. They take the brunt of the damage. Ingestion leads to catastrophic, irreversible liver and kidney necrosis. The organs literally melt from the inside out.
Oh my.
Without immediate, extreme medical intervention, which frequently requires a full liver transplant, amatoxin poisoning is highly fatal.
The sheer, terrifying risk of confusing a rain-washed Pholiota aurivella with a deadly Gallerina should be more than enough to keep this golden mushroom completely off the culinary menu.
I completely agree.
When you lay it all out like that, it's sobering.
Mhm.
If we pull back and connect all of these distinct pieces to the bigger picture, it really frames exactly what Pholiota aurivella is and what it isn't.
Absolutely.
It is absolutely not a culinary prize. It is a highly specialized master of forest recycling. It's a localized, acidic chemical refinery capable of digesting the toughest structural concrete in nature lignin. And while it's doing that vital ecological work, breaking down the forest so new life can grow, it's defending its fleshy body from an invisible swarm of bacterial invaders using a completely unique divergent arsenal of surfactant lipids, popping cell walls with linoleic acid, and deploying complex carbohydrate decoding lectins like PAA.
It really forces us to completely shift our perspective. We can no longer view a rotting log just as a piece of dead wood. It is the forest pathobiome. It's an intense, microscopic chemical battleground where every single organism, no matter how passive or pretty it looks on the outside, is deploying incredibly sophisticated biological weaponry just to carve out a few days of survival. Pholiota aurivella is perfectly, beautifully evolved to win its specific battles in that war.
Which leaves you, the listener, with a final provocative thought to ponder the next time you are walking along a trail through a damp autumn forest. When you see a splash of yellow on a log, think about the invisible machinery operating just beneath the surface. If a simple, slimy, wood-decaying mushroom evolved to completely abandon the traditional antioxidant armor of its ancestors in favor of synthesizing a membrane-bursting biological detergent just to survive the bacteria on a rotting log, what other completely divergent, totally alien chemical defense systems are quietly operating in the fungi right outside our doors, completely invisible to the naked eye?